As we wrap up this unit on DNA and gene expression, we're going to spend some time here in this last video looking at a couple of different sources of variation, or how we get differences in different genetic material. The key points in this video are going to look at how changes to DNA, what we would call mutations, are able to impact the structure and function of proteins. And then we'll look at how different cells in a multicellular organism can express different proteins, even though they all have the same genome. That's a process that we call differentiation. We'll start with mutations. But in order to understand mutations, we should go back and once more look at this idea of biological information flow and how it works. We start with the information in DNA, which is transcribed into the information in RNA. And then that information in RNA is translated into protein. And so it may be clear to you how changes at the DNA level can lead to changes in protein structure. If we change bases in the DNA, that's going to affect the sequence of the RNA, which is in turn going to affect the structure of the protein. These are what we call mutations. There are different types of mutations. We'll look at a couple of different examples. We can change an individual base of a DNA sequence. That's what we call a point mutation. Point mutations are also known as substitutions, and the effects of those point mutations can vary widely depending upon the particular nature of the actual mutation. So in this diagram, we see the initial sequence without a mutation. It's at the DNA level, it's TTC. Depending upon the nature of the change, that mutation can have different effects. It can be a silent mutation. Changing TTC to TTT takes an RNA codon of AAG and converts it into AAA. But in both cases, it's still calling for the same amino acid, which is lysine. So there is no effect of that mutation on the overall structure of the protein. The point mutation can be nonsense. What we've done here is we've changed TTC to ATC, which is instead introducing a stop codon, which will cause translation to stop at that point and will lead to a protein that's probably not very functional at all. Or it can be a missense mutation. We can change the codon to a codon for a different amino acid if that amino acid has similar chemistry to the chemistry of the initial amino acid. So for instance, lysine and arginine are both basic amino acids, so changing a codon for one to the other will probably not have as big an impact on the overall structure and function of the protein. Whereas if we were to change it from lysine to something like threonine, which is a polar amino acid and has a different interaction with the overall structure of the protein, that can have a pretty big impact on the overall function of the protein that's being built. Another thing that can happen at the individual sequence level is the insertion or deletion of bases, what we would call indels. These are going to lead to what are called frameshift mutations. What we see here is in the normal situation, we have a DNA strand that's leading to the production of a particular mRNA, and we can see all the amino acids that are going to be in that gene, starting with methionine and going all the way through to proline. What's going to happen in a frameshift mutation is we're going to insert a specific nucleotide into the strand here. As a result, our RNA is going to be impacted, and every codon downstream from that RNA is going to be impacted as well. Whereas before, it was glycine, tyrosine, alanine, leucine, all that way. Now, everything after that insertion has been changed, and we've actually even wound up introducing a premature stop codon as well. In other words, we've altered the reading frame of the gene, and that has had pretty profound impacts on the overall structure of our protein. We can also have changes to large-scale pieces of chromosomes, what's so-called chromosomal mutations. You can see a bunch of them over here. We can delete information or duplicate it or invert it, turn it upside down. We can take pieces of chromosomes and stick them on other chromosomes. We can see all these different possible changes that can result from these massive reorganizations of chromosomes. So mutations are not just limited to individual changes at the base pair level. They can also affect large segments, many millions or hundreds of millions of base pairs of the genome, depending upon the nature of the particular mutation. In considering the effects of mutations, it very much depends upon the particular nature of that mutation, as well as where it occurs in the genome. Many mutations will just be neutral in their effects. If we look at the entirety of the human genome, only 1.5% of our genome codes for proteins. As much as 26% of our genome is introns, which would be removed from those transcripts before they were spliced together to make proteins anyway. So if a mutation occurs in an intron, it's not going to have an effect on the structure of the gene. But additionally, we have a variety of other elements as well in our genome that do not contribute to the overall function of a protein. So when most mutations occur in our body, they actually have no impact on our proteins or on the traits that we show as a result. which is a good thing because mutations occur all the time in our genome when we're exposed to harmful chemicals and substances in our environment, or anytime that we replicate our DNA for the purpose of cell division. Leaving aside environmental drivers of mutations, every time we copy our DNA, the rate of mutations in our genome is about 1 in every billion base pairs that are replicated. Since the human genome is about 3 billion base pairs in length, that means every time a cell divides, we're going to get about 3 new mutations per generation of cells. This can't be avoided. It can't be stopped. So it's a good thing that much of our genome actually does not contribute to proteins and traits. Otherwise, our genome would mutate way too rapidly and the structures of those proteins would change too quickly in order for us to remain alive and functional. Of course, sometimes mutations occur that do have negative impacts. This chart shows some of the major mutations that are involved in a variety of disease conditions. For instance, we can see that in sickle cell disease, we're changing a glutamic acid codon to a valine, or in prostate cancer, we're changing alanine to a threonine. In most of these cases, you can see that the changes that are occurring are changing an amino acid with one particular kind of chemistry into an amino acid with a different kind of chemistry. This is going to have a big impact on the structure of the protein that's produced and on the function of that protein in the cell. But mutations can also be positive. Mutations are ultimately the source of all of the variation that we see in living systems. Every difference between you and any other human on the planet is ultimately the function of mutations. If the DNA had not changed, you would not have the variations that you have that make you different from every other person on the planet. This is going to be really, really important later on in our course when we talk about evolution. In order for evolution to function and allow populations of organisms to adapt to changes in the environment over time, you need to have a basis of variation through which evolutionary selection can operate. If you don't have variations present that can survive a particular selective change, then the population will go extinct. So even though most mutations are neutral, and many mutations can be negative, it's not correct to think about every mutation as negative. Some mutations have given rise to beneficial variations that have given rise in turn to organisms like you and me and every other organism that you see living on the planet today. The other major way in which variation is produced in multicellular organisms is through the process of differentiation. Starting from a single cell, your body has given rise to a variety of different cell types, which is interesting because every cell in your body shares the exact same genetic material, the exact same genome. So how does this differentiation occur? How do you go from that single zygote, shown here as a stem cell, and produce all the different types of cells in our body? The way in which it's done, it goes back to this idea of how eukaryotes control protein synthesis. In order for transcription of a particular gene to occur, you need to have particular transcription factors present in the regions of the genome that surround that particular gene. If you don't have those transcription factors present, you will not get transcription of that gene. And this is the key that explains differentiation. By having different transcription factors present, we get differential gene expression, which is what leads to differentiation. Even though every cell in your body has the same genome, every cell in your body is expressing different genes, and as a result is making different proteins, and so has different structures and functions. The expression of genes in a nerve cell that allows that cell to become a nerve cell is different from the expression of genes in a blood cell, or in a bone cell, or in a skin cell. Every cell type in your body has a different expression profile of the genes in the genome. And it's through changing the expression of these genes by changing the transcription factors that are present inside of each of these cell types that you wind up with the different types of cells that you find in your body. As we wrap up this video, when we look to check our learning, let's make sure that we can do the following things here at the end. Make sure that we can explain how different types of mutations can impact the structure of genes and the structure of proteins. You want to make sure that you have an understanding on how substitutions and frameshifts both impact the overall structure and function of mRNA and proteins as a result. Also make sure that you can explain how cells in a multicellular organism differentiate into different cell types, even though they all have the same genome. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions that you have so that you can get the answers that you need. Thanks again for watching this video. I really appreciate it. Have a great day.